of all, congratulations. Most people working in data science, for good or for bad, work for Google, Twitter, and so forth. And their real job is to make people click on ads. And by working with AI and ag, I think it's something a bit more socially noble, a bit more interesting. So I appreciate that. Again, congratulations. Let me tell you the format of this tutorial. This tutorial has almost no math or code. Who cares? I want to give you the intuition behind the state-of-the-art in time series data mining, give you pragmatic tools that you can apply, and literally in the first day, first hour, get interesting results on some important problems in this space. These ideas are very general. We've applied them to Mars rovers and to industrial machines and all kinds of things, but they do really work in ag and livestock settings. I personally have applied them to chickens, insects, marine mammals, cows, penguins, mice, and so forth. And the rest of my colleagues and collaborators have applied them to even broader animals. You might be surprised to learn there's no deep learning in this tutorial. I'm personally a deep learning skeptic when it comes to the case of time series. This might surprise you if you read the literature in this space. I don't have time to develop this argument, but if you want, you could look at this online tutorial that explains why I'm skeptical. So let's preview what we're talking about today. Let's assume you have some livestock time series data. What do you want to do with it? And the answer is everything. You want to classify, cluster, summarize it, visualize it, segment it into regimes, look for anomalous regions, look for conserved behavior, look for rules, look for emergent trends, and so forth. And my surprising claim is there was one simple tool, the matrix profile, that basically lets you do all the above and in a very simple and fast way. So that's the main take home today. Another important point I make today is that there are really two different situations that we're going to encounter when we look at this kind of data. One is when features and when behaviors are conserved in the shape space, and the other is when behaviors are conserved in the feature space. And there's almost no overlap between those two cases, but they're both easy to handle, again, as I'll show you. So there's one primitive we have to understand before we begin, and it's a very simple and obvious one, Euclidean distance. Suppose I give you these two time series. They could be two heartbeats or two gait cycles or two whatever it might be. And I ask you the question, are they similar? I think you can just look at this vision and say, well, C here is a bit noisier and it has a different angle here. It's a bit taller peak, but in general, they are reasonably similar, I think. But we want to quantify that mathematically, of course. And there are at least 100 different measures for time series similarity, but the oldest one, literally thousands of years old, is probably still the best. It's the classic Euclidean distance. Here's the formula for it. I'm sure you've seen it before, but the intuition is very obvious. We're going to take the two time series, Q and C. We're going to align them on the same level. And then we can look at the pairwise distances between the ith point and the ith point, the jth point and the jth point, and so forth. Those pairwise distances could be negative or positive. So we're going to actually square them, that's this here, make them all positive, sum them all up, take the square root, and that is the Euclidean distance. For those of you who prefer correlation, it's logically equivalent to Pearson's correlation. It's important to recognize actually that we're going to z-normalize the data before we actually measure the Euclidean distance. So the distance between here, Q and C, is the same as the distance between C and Q2 or Q4 or Q3. Those other time series have different levels and they have different standard deviations, but we remove that before we make the Euclidean distance calculation. So I think I can show a picture of that. Here we go. We would basically slide the time series to have a mean of 0, and then we divide by standard deviation to have standard deviation of 1, and then compute the Euclidean distance. And that's almost always the right thing to do for almost every data set. And of course, it's trivially easy to do. It's one line of code in MATLAB, for example. Okay, with that little background, we're now ready to actually get into ag. So we're going to use our friend Apollo here to make a running example of this tutorial. And our friend Apollo has a sensor on his neck, and that produces time series. In this case, it produces time series at 50 hertz. 50 hertz is pretty good for a large mammal, like a human being or a cow or a goat, potentially. I worked with chickens. Chickens actually, because they're smaller, can move faster. And you might be at, say, 250 hertz for chickens. I worked with insects. Insects are very small. They have to be at over 1,000 hertz. But 50 hertz is perfectly fine for our friend, the cow. And the sensor produces, at the triaxial accelerometer, it produces X, Y, and Z. This is a few minutes of time series. So it's important to realize that in general, when you get data, X, Y, and Z have no intrinsic logical meaning. They do for ships and for automobiles, there's a standard meaning, but for animals, there isn't one. What you really want in any case in a biological sense is surge, heave, and sway. And you can map these onto X, Y, and Z. They may not map on originally exactly, but that do some adjustment basically, but these are the standard notations for all animals. As it happens, for many problems, it turns out it makes basically no difference. You don't really care which is which. And we'll see that in a moment. I want to mention in passing that actually, although I've mostly worked with accelerometers in the past, recently markerless tracking of animals has become incredibly robust. And of course, it's very cheap and easy. This particular software package, Deep Lab Cut, is really amazing. It works on fish, insects, amphibians, reptiles, cows, whales. It's an incredibly general and simple package. I highly recommend it. And this will, of course, produce time series from this cow and so forth. For this tutorial, I don't care how you get your time series. It's all the same to me. So here we have our friend Apollo. He's given us about 1.2 hours of time series data. Now begin to play with it. Just one piece of advice: when you're going to get this data, try to get as much labeling about the data as possible when you record it. So for example, here we have the label of some data from Dante, some data from Apollo. We might also have labels that some data comes from Apollo before an operation, and some of the data comes the next week after the operation, or some data is one control, like Apollo on pasture versus Apollo on grain, and so forth. So just try to get all the labels when you collect the data, including labels like when you collected it, was it warm or cold, was it sunny or overcast? Basically, any bit of metadata you can get should be recorded, because a year later when you're recording, or so you're analyzing this data, you really might want to know some conditions that existed. As it happens, this data set has really beautiful labels. Some human being watched these cows and actually recorded and labeled the data second by second into these 3 different behaviors: grazing, ruminating, and rest and other. And again, labeled data is worth its weight in gold, basically. Labels are expensive generally. Of course, not all the data has to be labeled. If you record 10 years of data, it'll be very expensive to have a human label all of that. But the good news is that if a human can label, well, a small subset of the data, you can often brush those labels onto the rest of the data. And I'll explain how to do that in a moment. So this data is too big to actually see easily. Let's zoom into a little bit of the data and we can analyze this. So here I have a small subsection of the data. It's about 20 seconds long. I'm going to call it Apollo Y subset or sub. And this is some data. And it's not visually obvious right now what this means in anything, right? But let's see what we can do with it. So I'm going to say for the moment that I have offline got a small subsequence here in blue. I'm going to call it query. And how I got blue we'll see later on. But essentially, this is some bit of data that I have reason to believe is indicative of mad cow disease, maybe some asymmetry in gait and so forth. So my hypothesis is that if another bovine has this shape, they may also have mad cow disease. So given that, my question is, does this query behavior exist somewhere within Apollo? When you ask a matching question in DNA or text, you can get exact matches. But of course, with real value time series, you never get exact matches. You just get very close matches. I'm going to define closeness by, of course, Euclidean distance. So here's what we're going to do. we're going to build what we call a distance profile. A distance profile is a real important workhorse in all time series analytics. What I'm going to do is I'm going to take the query, I'm going to measure its Euclidean distance to the first part, the prefix of this red time series. Just like I did here, and I'm going to write down here with this blue dot the Euclidean distance. It was about 12.2, I guess, here in this case. I'm now going to take the blue query time series. I'm going to shift it over one click. I'm going to remeasure the Euclidean distance. This Euclidean distance is a little bit smaller. It's now 11 point something. I have a blue dot that represents, sorry, a green dot that represents that here. If I keep on doing this and slide the query all the way to the end, I create this green shape here, which is called the distance profile. So to be clear, the red data is real time series from a real cow. The green data is like a meta pseudo time series that really's job is to annotate the original data. The distance profile is almost the exact same length as the red time series. It's a little bit shorter because the blue query bumps up against the end and it can't go any further. So how do you visualize and interpret the distance profile? Well, it's quite, I think, obvious. If you want to find the best match to the query, you simply look for the lowest value in the distance profile. So the lowest value is right here. And at the location of the lowest value, if I project up, that subsequence here I'm claiming is the best match to the query. And in fact, I've overlaid the query so you can see that. Notice actually that it's a bit stretched in the y-axis compared to the query, but that's allowed because we're doing this again in z-normalized space. So I now know the location of the best match. Here it is here. And I know the distance to the best match is simply the height of the green curve, which was 1.26. Naturally, I can now go and get the second best match. That's going to be the lowest value, not including this. My guess is probably that guy here. And indeed, that is the case. Now I have the second best match. And of course, I continue this to find the next best match, probably this guy, and then this guy, and so forth. Eventually, I run out of good matches, but I can keep on doing this to find the K top best matches. So one important point is here is the absolute value of the numbers, 1.26, have no intrinsic meaning. Only the relative values matter. 1.26 is a lot less than 10, and it's a lot larger than 0.0001. If for some reason you needed to have the number to have meaning, you could convert this to Pearson's correlation. People do this all the time, for example, in seismology. But for us, it turns out we almost never care. We only care about the relative rankings to do classification and clustering and all those other interesting things we want to do. So I'm going to show you a worked example of this, show you the real power of this very simple tool to solve interesting problems. I have this gray time series here, and this actually is from the hip of a dog walking in my lab. And it's annotated labeled data, that's good news. The dog was walking originally on concrete, then the dog was walking on carpet, then the dog was walking on concrete again. And it's not visually obvious on this, but my claim actually is that that might have changed the gait of the dog a little bit, and that might be a recognizable fact. So I have from out-of-band information, a query. Here I'm plotting it at a slightly larger scale so you can see it. And I know this query comes from my same dog. The question is, do you think the query comes from walking on concrete, or does it come from walking on carpet, or maybe something else completely, like walking on grass? And we can solve this simply by doing what I did in the last few slides, which is to compute a distance profile and find the nearest neighbor to that query. And here I've done that. So the nearest neighbor I'm showing you right here in context is because the lowest part of the distance profile, you can see the very low value here in green. And because the nearest neighbor to the query is in the carpet region, I'm guessing my query might have actually come from when the dog was walking on the carpet. You can see actually the best match here is not perfect, but it is really, really good. That was just one example. Let's try it again, make sure. Let's actually find the top 16 matches. and the top 16 matches all occur within the carpet region, then I have very strong confidence that my query belongs to dog walking on carpet. Before I move on, this is actually a quite surprisingly useful tool here, because in a sense, I've done something even greater. I've done semantic segmentation. So if I did not have the label concrete, carpet, concrete, and I just looked at where the matches are, they begin here and they basically fill the space and end here. And that strongly suggests that this is a coherent segment, a coherent regime. And dividing data into regimes that have semantic meanings is a really important and useful tool. So this very simple tool can be very useful in many situations. I'll just mention in passing that this is surprisingly fast. This takes less than 1/10,000th of a second to do. So computationally, it's incredibly cheap to do this. So let's just do a quick mini review before we move on. The simple idea is called query by content, or similarity search, or nearest neighbor search. And we can use this tool, the distance profile, to find the k best neighbors to any query you want to give. And I previously hinted at, this is a simple but incredibly powerful and useful tool, limited only by your imagination. So I've actually computed this distance profile more than a few trillion times on more than 1,000 different data sets for different customers or problems it might be. And it's amazing how often this thing actually solves a problem or gives you a big hint as to solve the problem at hand. It's simple but incredibly powerful. It is, I think, the most important subroutine in all of time series data mining. You might say, well, my example actually has one dimension, the y or the x, or something like that. You have, high dimensional data, x, y, and z. Very good news. You can simply compute the individual distance profiles, send them together, and then look for the smallest value, and that is the multidimensional nearest neighbor. So this generalizes trivially to multidimensional data. I have you a full worked example in the appendix of my slides. As I previously hinted at, this is unbelievably fast to do. So let's say you have 24 hours of our friend Apollo's bovine data. That's about 4.3 million data points. We could search that in well under a second, even on a cheap laptop machine. The speed at which we can do this is due to an algorithm called MAS that was invented by my former PhD student, Abdullah Mwin, who's a bit of a genius. And you can get MAS in basically any language, any platform these days. Okay, we're now going to see a more general tool that sounds similar, but is a little bit different, called the matrix profile. And I'm going to modestly claim the matrix profile is the best idea in data mining in the last decade. It's a very simple, almost trivial idea, but my claim actually is, once you compute the matrix profile, then almost all problems you want to solve, classification, clustering, motif discovery, anomaly detection, rule discovery, blah, 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 become trivial. They're essentially solved for free. This is a very brief introduction to the matrix profile. If you want, there are longer tutorials online by myself and by other people too. There's a danger it's going to look a bit vainglorious because this came out of my lab, but I do want to say that actually that matrix profile is widely used now. There are many hundreds of papers, people use it to solve all kinds of problems, literally in outer space, but in all kinds of medical, biological, anthropological, industrial, social settings. It's a very well used tool these days. Okay, let's return to our bovine example. And here is Apollo Y sub, our small subset of time series. And now what I'm going to do is to illustrate this, I'm going to pick a random location. I have to pick 215. Here's the time series subsequence at that location in pink. I'm going to find its nearest neighbor in the rest of the time series. How would I do that? Well, actually, I could use the distance profile we just saw, but I don't care how you do it. All I care is to find its nearest neighbor. Its nearest neighbor might be, for example, over here, or it might be here, it might be here. I don't really care where it is. All I care about is how far was it in Euclidean space? And the answer you can see is it was 6.1 units to its nearest neighbor in Euclidean space. Once I know that, I'm going to put a little blue dot at that location. And so if you see this blue dot, it tells you that the subsequence here has a nearest neighbor somewhere, we don't care where, whose distance is 6.1. Let me do it again with a different random location, this time 5, 4, 7. Here's the subsequence. I again search for its nearest neighbor somewhere in this red time series, and I find its distance to its nearest neighbor is closer. It's 2.2. It's a small value. I put a blue dot right here. Now let's imagine I do this for every single location in my Apollo Y subsequence. What would the blue dots look like? They look like this. This is called the matrix profile. So the matrix profile simply encodes for every subsequence how far it is to its nearest neighbor. And again, it's a very simple idea. My claim actually is that this matrix profile has metadata that will let you solve almost any problem you want very simply and easily. So there are a couple of locations of the matrix profile that have a special name. Let's actually know those names. So I look for the highest value. It's high here, but the highest value is right there. The highest value has the name time series discord. And what that tells you is that the subsequence of this location here is in some sense the most unusual, the most unique, the most novel, the most anomalous subsequence in all the data. Another place that's named is the lowest locations. As it happens, they have to be a pair by definition, and the pair of lowest locations, here and here, are called the time series motifs. And this pair, here in green and here in cyan, look the same by definition. They're the most conserved subsequences in all the data. So this actually might be a repeated behavior. Maybe literally the cow sneezed here and made a shape, and the cow sneezed here and made a similar shape. It was conserved. But there's probably some causal reason why these two things are so similar. It would be very suspicious if that wasn't the case. So this is so important. Let me say this again and remind you how to read a matrix profile. Here I'm showing you a matrix profile and deliberately not showing you the time series. So we just zoom in and focus on the matrix profile. And so if you see a matrix profile and you see low values like here and here, that tells you that the corresponding time series at that location has a conserved behavior. It doesn't tell you where it was before in time or after in time, although we can get the information elsewhere. But it simply says that when it was here, its behavior was repeated at some point in the past. Likewise, if you see a high value in the matrix profile, it tells you that the behavior at that location is either novel, unique, noisy, anomalous, strange, different. It's not typical behavior. So now you know that, we can now practice our skills at using the matrix profile on some data sets. Let's start with this data set, which is the vocalization of a zebrafinch, or I guess zebrafinch. And my question I'm asking you is, can you see any conserved behavior in its vocalization? So I guess this is kind of conserved, but it's kind of boring because it's flat. And that looks a bit like this, maybe, and so forth. It isn't easy to answer that question, but if I compute the matrix profile and plot it next to the data, it becomes trivially easy. I can simply look for the smallest values of the matrix profile, which are here and here, and that corresponds to that shape and that shape, which I plotted here larger scale, and I've called it motif 1. Actually, there are 2 more examples very much like that in this data set, so I've actually put them here in gray. Once I've found motif 1, I can now go back into the matrix profile and look for the next best conserved motif, which of course is motif 2, which is the shape here. And then go back in again, look for the next most conserved motif, which is motif 3, which is this guy here, even better conserved visually in my mind. And there's more examples here shown in gray. This actually hopefully should kind of excite you a little bit, because it tells you there's a lot of conservation in the zebrafinch's song. It's not a random utterance. It has some structure. And actually, if we took the time, there's some evidence of a vocabulary, which is kind of obvious, but maybe even a grammar. What I mean by that is maybe when you see motif 2, then within a half second, you tend to see motif 1, but you'll never see motif 3 after motif 2, and so forth. And by using things like Markov models and so forth, you could actually potentially find these grammars. So all I've done here is find conserved structure, but downstream algorithms could actually process this to do other interesting and cool things. And I did this with the matrix profile. This is an example of finding motifs matrix profile. For my example of finding anomalies, I had to switch gears to a non-livestock example just because I want to have a data set which you have good intuitions. So this is the taxi demand in New York for 3 months in 2014. And what you see actually is, what you might think it is, this is a daily cycle here. So the lows are, I guess, 3 o'clock in the morning, no one's using the taxi. And then you have a morning rush hour, people are busy going to shows and dinner at night, and it goes back down to close to 0 in the middle of the night. And another pattern you can see here potentially is, if you look carefully, is there are 2 out of 7 days that are lower than the rest. They correspond to Saturday and Sunday. So those are patterns you could see with your naked eye, but the question is, are there any additional structures or patterns in here? And once again, the answer to that question we can find quickly with the matrix profile. Let me compute it. Here it is. So the matrix profile is mostly low values, which makes sense because most days are like most other days in taxi world. However, there are some exemptions. So here is a strong peak, and that suggests that the values here are different, unique, anomalous, or strange. As it happens, this corresponds to Thanksgiving. So it's rare we see a reduction in taxi demand on a Thursday in a busy city, but this is because of Thanksgiving. The next highest peak here doesn't correspond to an American holiday, so that confused me at first, but then I realized what the issue was. This spike here actually is a formatting error. So this is actually daylight saving time, and 2 hours of data were compressed into 1 hour in the spreadsheet, make it look like there's a big burst of taxi activity, which there really wasn't. So I found a second anomaly here. And the last anomaly I note here is very subtle, but nevertheless, these days are a little bit different to the other days based upon the height of the matrix profile. What caused that? Well, actually, this is actually Columbus Day. Columbus Day is basically ignored in most of America, but in Boston and New York, there's some Italian Americans who still celebrate it. And so they actually have parties and whatnot, and that changed the traffic patterns right here. So again, this is kind of exciting. This is a data set. It happens to be small here, but I can do this on 1 million or 1 billion or 1 trillion data points. And I can find very subtle anomalies simply by looking for the high value matrix profile, then zooming in the data and saying, why are you different? Why are you unique or strange or anomalous? And that can be incredibly interesting to do. Great. Let me put this all together with a kind of a worked example over the next 6 slides with a marine mammal. So here we have a nice data set from a marine mammal, and it's labeled, not perfectly, but quite well, by a grad student, both side, paper and pencil. And so the question is, there's obviously some kind of structure here, but what is the structure? Can we exploit it? Can we understand the structure in this data set? And of course, the way we're going to find structure is, you guessed it, compute the matrix profile and look for motifs. So this data set actually is very rich in motifs. So there are 4 main ones here. A, here are 2 examples. B, here are 2 examples, and so on. Notice motif discovery is quite robust to noisy data. So for example, one occurrence of A has a dropout here, but the algorithm can actually find that not a problem. As I previously hinted at, by labeling these things A, B, C, D, we can actually now reason at a meta level about the existence of these patterns. And we can ask, is there a meta pattern in the ABCDs? And actually, I found one instantly, which is the following pattern. If you see the pattern D, then you tend to see the pattern C almost immediately. So this is like an antecedent. This is a consequent if you think in the logical language. So here's what this actually looks like. Here are 2 examples. here's the motif D that occurs. And then in the first case, almost instantly, we have motif C. In the second case, there's a small delay of time, but not much. And we see the motif of C. So this really suggests there's kind of a rule. And what does it correspond to? So the flat section here is the seal basically foraging at a uniform speed. But at some point, he sees a tasty meal, a fish, and he has a burst of acceleration, as we have seen right here. He actually manages to catch the fish, but by definition, he's going to catch the fish tail first. It's very hard to swallow a fish tail first. So the seal is going to rise to the surface and juggle or flip the fish up in the air, catch it again by its head, and eat it. And this is this behavioral motif we discovered here. So before I move on, this is an incredibly useful and powerful and general trick. If you can look at your data and you can find motifs and give them labels, A, B, C, D, E, F, G, you can now use other kinds of algorithms in text processing or association rules to find meta patterns within that data. So you can ask questions like the following. Suppose you see 10 B motifs per hour in males, but only 1 or 2 B motifs per hour in females. Why is that? Why are they such a dimorphic in behavior? That could be an interesting question to ask. You can also ask questions based upon your interventions. You could feed your cow some kind of feed for a week and look at the motifs, both the shape, the occurrences, the time of occurrences, morning, afternoon, and so forth. Then you can change the cow's feed and see if the motifs change also in shape, occurrence, time of occurrence, and so on and so forth. So again, I've done this in dozens of domains, in industrial domains, medical domains, and agricultural domains. It's an incredibly simple but incredibly powerful technique. We often find very useful, interesting, actionable patterns by its motif discovery. So let's return to our friend Apollo and do this on Apollo. I'm going to compute the matrix profile for this entire data set. Notice to do this, I have to compute the pairwise distances, this Euclidean distance right here, between all possible pairs of subsequences. And there are basically 24 billion such pairs. It's this number squared divided by 2. And you might think computing this Euclidean distance 24 billion times would take a long, long, long time. And that was true for the invention of the matrix profile and the algorithms to compute it. With the matrix profile, we actually compute this in just a few seconds. So much, much, much faster than real time. And so when I did this, I found the best motif happens to be a pair that occurs very early on in Apollo's day. It's this blue and red pair you see here. And you can see these actually are very, very strongly conserved bits of data. Now, I noticed actually that both of these pieces of data occur in the same semantic behavior, ruminating. And I also noticed actually they're very similar. In fact, they're exactly 1.29 units apart. And so this, to me, suggests technically a decision rule. And here's the decision rule I made up based upon this. I'm going to say that for the rest of Apollo's day, maybe for the rest of Apollo's life, if I see a subsequence X, such that the distance between one of these motifs, the blue one here, and X, is less than the 1.2 times 3, I can now claim that the bovine is ruminating. So just quickly, where does 3 come from? I kind of pulled it out of the air. It's an old trick in statistical process analysis. I could tune this to make it more liberal or more conservative, which would change the precision and recall, but it's basically a reasonable number I pulled out of the air. So my claim is I can actually run this rule on the rest of the data and try to find out when this bovine is ruminating, if I didn't have the labels from the grad student. So I tried this. Here's a zoom in on the first part of the data so you can see it more easily. Here's my decision rule. This is basically 3 times 1.29. And what we actually see is it works quite well. So in the first section of ruminating, we have lots of hits that are smaller, telling us that this is a ruminating section. And then this small region here in white, where there was no ruminating behavior, has no examples below the threshold, correctly saying these are true negatives. Then I have another burst of ruminating here, lots of examples below the threshold. I have another correct example of a true negative, and so on and so forth. And then for the rest of the time, when it's not ruminating, I have no false positives except for one. It might be a genuine false positive. It might actually be the grad student missed this because it was so short, but this is actually quite nice. This incredibly simple rule that I made in 2 seconds of work is correctly segmenting my data set. And just quickly, it's always good to actually plot data and look at data, because occasionally you do see meta patterns you didn't expect and so forth. Here's a tentative one that may not be true. I noticed actually that the ruminating behavior is not uniform in this section of time. There appears to be kind of a burst of behavior, then a relaxation, a burst of behavior, relaxation, burst of behavior, and so forth. Kind of a meta periodicity. I don't know if this is actually true or not, but it's kind of suggestive. It's worth actually looking at offline. Okay, before I move on, I want to say actually, this is kind of amazing. All right. I know nothing about cows or bovines, and I only typed about 8 lines of code. And I used about 1 minute of my brain power and a few seconds of CPU power. And doing that, I was able to correctly annotate the complex behavior of a bovine. So this is actually really very, very promising. Simple ideas, simple code, very little CPU time, you could potentially extract interesting, actionable, useful stuff from your chickens, your cows, your pigs, whatever it might be. So you might say this is a fluke. Let's try it again. This time I'm going to do it with chickens. I actually have 4 years of chicken data, not one chicken, many chickens in parallel. And I use multi-discovery to find a template. Here's the template here in purple. And it happens to be associated with a behavior called dust baiting. So I'm going to search for this template in my 4 years of data set, which is about 12 billion data points. It's a pretty big data set, but we can search it easily. And I look for the 1,000 nearest neighbors. So here's the template. Here's the best match. It looks almost identical to my eye. And the second best match looks almost identical, and so forth. In fact, the 1,000 best matches all look really, really identical. This behavior of dust baiting is deep, deep in the chicken's reptilian brain. I think it's co-opted from when the chicken evolved from a fish, basically. So it's an incredibly well-conserved behavior. And this is actionable because as it happens, my chickens are divided into 2 possibilities in different parts of the farm. One is a set that actually is known to have mites, and the other is a set that is known not to have mites. And I can now ask the question, is this behavior more common in the mite birds or the non-mite birds? And the answer is it's much more common in the has mites class. This means I now have basically a predictor or a proxy for mite infestation that I could actually discover with a camera or some other test and take interventions and actions with that. And once again, I did all of this with less than 10 lines of code. I did it in less than a few minutes CPU time. I searched these massive data sets, and I got a useful, actionable rule out of this. So I'm not going to show you this today, but the matrix profile actually generalizes even further. I showed you an example where we actually look within one time series, but you can look between 2 different time series. These are called joins. We can actually use this then to compare and contrast behaviors. So you can ask questions in English and then translate them into 2 lines of code and solve them. Those questions in English could be things like, what patterns occur in males but not females? Or what patterns occur in both Hereford and Holstein cows? Or in time, what patterns occur before the Newton but not after the Newton? And so on and so forth. In addition, I want to point out actually that this matrix profile stuff, once you install it on your machine, is just 1 line of code you have to call to get all these information. And as I hinted at earlier, there's a large and growing community of matrix profile users, so you can get these tools in every language you want very easily free in the web. So I'm going to stop here at the matrix profile. There are some other primitives that we can use. We've seen motifs, we've seen discords, but it's also chains, novelettes, shapelets, Plato, snippets, and floss, which all have slightly different uses and meanings, which are equally easy to use and equally fruitful, but I won't show you those today in the interest of time. So I'm going to switch gears just slightly. There is some bad news here, which is sometimes the behaviors that are of interest to us are not well captured in shape, but only in features. Let me explain that by talking about humans instead. So human behaviors are often captured in the shape. So for example, there's a shape of walking, running, swimming, cycling. They all have strong dynamic shapes. And actually, you can look at the difference between walking upstairs, walking downstairs with an accelerometer and classify those very well. So dynamic behaviors really exist and work well with shape, and therefore work well with the distance profile and the matrix profile. These are all shapes here we can see very nicely. When I cross this threshold here, I now move to a region where there are no real shapes per se, but only features. So going back to our human example, think about watching TV or reading or using the computer or sleeping. There's no dynamic shapes there, but there might be some kind of features in the data that correspond to that. And so we have to, for some problems, abandon the possibility of looking at shape, but look at features instead. So here's how this would work. It's quite similar. We would have subsequences of data. Here's subsequence highlighted here. But instead of actually reasoning about the shape of the subsequence with a matrix profile or distance profile, we would simply pull out a feature vector. We'd grab this subsequence and we'd measure its maximum value. Here it is here. We'd measure its minimum value. Here it is here. We'd measure its entropy, its zero crossing rate, its skewness, its factor dimension, and so on and so forth. And now, once we have a feature vector, we can actually use all the classic tools that are out there, nearest neighbor, decision tree, naive Bayes, and so on and so forth. However, there's one thing I left unspecified, which is which features should we use, right? Why is minimum and maximum good? Maybe they're not. So it turns out actually there are many features we could use. In fact, people have documented up to 9,000 different features, a very scary number. And of course, of those 9,000, many are probably useless in general, and most will be useless for your particular problem anyway. Furthermore, many will be redundant. You're basically measuring things twice. So what are the best features to use? And this problem was partially or maybe even largely solved by two guys, Jones and Fuckler. And they searched through all 9,000 features on over 100 data sets with a brute force combinatorial search to find a nice small subset that had the following property. They're mostly non-redundant with each other, and they do provide semantically meaningful discrimination on most problems and most domains. And they call the subset a stupid name, but nevertheless, catch-22. So actually, I do think that catch-22 is one of the best ideas, most useful things in data mining in the last decade or two. It's a really nice feature subset. It's kind of a one-size-fits-all, which is never true in general, but it is almost true in this case. And it's at least the best starting point for almost everybody, for almost every problem, rather than doing anything else in this space. Unfortunately, the names that they actually came up with for these 20 features are awful. They have no really good semantic value, but here are the names of the 20 features, or some of them here. And so my claim is we should begin by taking a subsequence, computing these feature vectors, 20 feature vectors from them, and reason with those feature vectors. Let's see a worked example. So here I have a behavior here. Let's call it class A. And now I'm going to compute its 22 feature values using catch-22. And I'm going to plot them here in this colorful bar chart. These features can be positive or negative when normalized correctly. And so once again, the absolute meaning of these things isn't particularly relevant, but I guess in this case, this class A has a high positive load here in this blue feature, which is whatever this thing is here. And class A has a high negative load in here in red. which is this feature here, and so on and so forth. And so you can think of this colorful bar chart here as being like a signature or a fingerprint for the class A behavior. And the hope is that if we encounter the class A behavior going forward, again, it'll have a similar fingerprint and we can recognize it. And we can recognize it, for example, by computing Euclidean distance between this signature and the ongoing signatures. Let's see if this actually works. So this, again, is a proxy for class A. My claim is we're going to reason with these things here and basically almost ignore the original data. And then any algorithm we have of interest, whether it be classification or cluster anomaly detection, we're going to reason in this space, not in the raw data space, because the raw data space may be useless, as I previously hinted at in this case. Okay, so here we have two unknown objects, unknown 1 and unknown 2. I'm deliberately temporarily hiding the time series from you. I'm going to ask you the question, do we think that one of these is close to or similar to class A? And so it's very unlikely to be this guy here, right? Because this guy here, unknown 1, has a negative reading for blue, whereas class A is a positive reading for blue. And it has a positive small reading for the red feature, but this has a negative reading, and so on and so forth. So these feature vectors just look so different. I suspect these are quite different time series. In contrast, unknown 2, the feature vector actually looks visually quite similar. There are some differences. This black thing here is quite different, but in general, most of the features are well conserved here. Let's fill in the picture, and now it actually kind of makes sense. Unknown 2 does look like class A, both in the raw space and the feature vector space. And unknown 1 does not look like the class A in either space. Before I continue, an important note here. In this particular example, uniquely, it's true that the shape is conserved as well as the feature is conserved. But more generally, that will not necessarily be true, which of course is the whole point or power of this. There are cases where the feature vectors will be conserved, even though the shapes won't be, but they come from the same semantic class. So again, as I mentioned before, things like dynamic behaviors, walking, running, and swimming, will be conserved potentially in both spaces. You could use either shape or feature vector. But low dynamic behaviors, in the case of humans, reading, watching TV, resting, sleeping, will only be preserved in this space right here. And of course, our cow has the exact same kind of possibility. Our friend the cow can be walking or running, which are quite dynamic, or it can be simply just lying in the field, which is very static, and will be conserved only in the feature space, not in the shape space. So once you have these feature vectors, you can do all kinds of interesting things. Let me kind of finish up today with a nice example of this. This is a fairly recent example from my lab. So we actually have these mice, which are models for Parkinson's disease and other brain abnormalities and dementia. And the mouse is actually asked to walk on a circular treadmill. And we're using that trick I mentioned previously earlier of markers capturing to capture various limbs as it walks on the treadmill. So we're going to build a catch-22 basically matrix profile. It's like the matrix profile, but not in the shape space, but rather in the feature space. And we're going to look for anomalies in the behavior of this mouse, maybe due to its cognitive decline. As it happens, there could be other anomalies that simply just due to formatting errors. If that's true, you should actually remove those from analysis anyway. So finding anomalies or errors is very important here. So if I begin this experiment, I look at the y-axis location of the right paw, you can actually see a gait, a gait, a gait, a gait, a gait. It's basically tracking the mouse, life is good. But we do actually find some anomalies, and we can find them easily with this little trick here. So this is the matrix profile in the feature space, and it's pretty low, but again, there's a big peak here. And this peak corresponds to actually a formatting error. Temporarily, the software confused the tail and the right foot, and it made the right foot apparently jump, when of course it really didn't, to the tail. So once we find this, we of course can remove from analysis. That makes the biologist quite happy. Another error we see that's formatting based is occasionally the tail will swing and it will occlude the foot, and we lose contact with the foot. And then again, we get a spike or an error. That's what we see here and here. But for fun, my favorite anomaly in this space is very occasionally the mouse gets tired of walking a treadmill and simply grabs onto the treadmill and rides it for a full cycle around. It's very cute to watch out as it happens. And when it does that, it produces this big and long anomaly here, D. And again, we have to discover those, move those to analysis, we give this data to the doctor. So again, if this is a few minutes of data, we could do this by hand, but because we have many tens of hours of data, this algorithm actually lets us automate the removal of anomalous data, and we only give the doctor clean data they can use to discover potential precursors of dementia. So I'm almost done here. Let me summarize. If you have data that has conserved shape, which is very common for all kinds of animals, then I claim that the distance profile and the matrix profile will solve almost all the tasks at hand. And that's true if you want to do classification, clustering, summarization, visualization, anomaly detection, rule discovery, you name it. It's a simple but very general tool. It'll solve almost all your problems and do it quite fast and easy. You may have data that has not conserved shape, but conserved features. And my claim actually is that the catch-22 with an appropriate algorithm will get you most of all the way. You can do a little bit better here sometimes. You can actually take 20 features and remove some occasionally that are irrelevant for you. And you can sometimes add a new feature that's bespoke for your particular problem. So maybe you know that for pigs, there's a feature that might be useful that isn't in catch-22, and you can add it into the set. But basically, once you do that, this will get you a long, long way. You probably understand that today I've shown you only single time series examples, and you often have multiple time series, roll, pitch, yaw, temperature, pressure, and so forth. The good news actually is that the distance profile and the matrix profile trivially generalize to those cases. No extra expense in time or effort, very easy. However, I will say, surprisingly, I've solved lots of problems in this space, and most of the time you can solve the problem with just one dimension, which is maybe surprising to you, but is true. And you can also use catch-22 multidimensional data, although it's less well understood and less common in literature, to be honest. And my second summary is at a much higher level. So I do edit and review for some of the best data mining journals and conferences. And I'm always disappointed at how rarely we see papers on livestock problems. People are doing great work on socially noble, important problems, but they tend not to send them to the best journals and conferences for some reason. So you can publish in those venues. I have done so, KDD and data mining and all these other places. So please consider sending your best work to the best venues. I think the world needs to see the utility of data mining beyond competition advertisements. As an aside, if you're going to publish, please share your data. If you have short questions, I think there's a small session at the end of this. We can answer questions. If you have longer questions, please feel free to ask me offline. I'd be happy to try and answer if I can. The slides are available if you want to use them for a class. And then finally, I just want to say thanks to all my great students for helping me make these slides and do this research. Thanks for the invitation to this session today. Thanks to our friend Apollo for giving us the data. Thank you for listening to me. I know you had a long day. Your day is ending, my day is beginning right now. And just briefly, if you're interested in insects and agriculture, I'm an expert in that. And I actually have both an academic interest and a small company that makes these wonderful sensors to detect and classify insects in the field. If you're interested in that, feel free to talk to me about that offline too. So with that said, I'm happy to answer questions if there are any.
Thanks so much, Eamonn. I'm sure we've had plenty of people that have really enjoyed that. So I've invited a few people to join a panel that I think are probably more interested in your work than maybe some others and might have some questions that other people will have. So I think Georgiana might kick things off. I see she has her hand up, so we'll let her go first.
Great. Hi, Georgiana. I met you before. I met you by email, so nice to meet you in person.
Hi, Eamonn. Thanks a lot for the great talk. It was really interesting and really nice teaching. Very great. I had a question about noisy data. Most of the data, especially from animals, I suppose, is very, very noisy. So how, do you use it as raw? Are there some pre-processing techniques that you usually do? How does it affect the distance profile?
Yeah, I mean, surprisingly, these techniques tend to be quite robust to that. So if you look at, Apollo here, there's definitely noisy data there. So actually, let me go back to this example here. So I would never, for example, say this is ruminating if 100% of the data is past the threshold. That's never going to happen. The collar's loose, there is some noise and so forth. So I have to have a meta rule that says if you have more than 5 examples of ruminating per minute, then the overall data is ruminating. So some extra kind of post-processing on top of that to take into account the fact that some of the data is missing and noisy can be helpful. You can smooth the data and so forth, but in general, distance profile, matrix profile are quite robust to noisy data that's intermittent here and there. If everything is noisy, it's not clear what you can do at that point, but at least occasional noisy data isn't a big deal in most cases.
Okay, thanks. I have a follow-up question, if my colleagues allow me. I was wondering if you looked at, I found it quite interesting that basically you can define a vocabulary using the matrix profile. So have you looked at using this as an explanation? So basically you create a vocabulary of primitives and then you use these primitives to explain to the user what's going on. And in relation to that, what is the redundancy of motifs? Because if you find, 1,000 things that all look the same, that they are basically one motif, how do you do it?
Yeah, good question. So yeah, we actually have done this. I actually haven't published this too much, but I consult a lot in oil and gas just because they have money. And they're interested in monitoring these enormous distillation columns. They have a 1,000 time series, and they want explanations as to problems or faults. They have a fault called foaming, for example, and they want to know what causes that, and they can find precursors. So if I can tell them, I've noticed that when you see motif X, within 10 minutes, you're going to have a foaming event, then they can actually monitor for this and then look for that foaming event and try to intervene and stop that event. So there is some utility like this, maybe not explanations, but at least using precursors to say, if this, then that. What's very helpful is if you have out-of-band data or annotations. So here I have this annotation of some grad students, and those really are very, very useful to actually provide that kind of explanation and so forth, right? But the more general question is to be looking at a motif and explaining it is a bit trickier in natural language. And people are doing stuff like this, but there's nothing really, I think, very strong and compelling at this point.
Thanks.
Explanations are the kind of what was the one for very, very desirable. We look at explaining things better, right? Because I often find things and I'm not sure if it's meaningful or true, and being able to explain it is clearly important.
All right. I have a couple more, but I'll keep them for later just to let my colleagues come in. Thanks, Eamonn. Great talk.
I'm seeing some come in the chat box. So Mark, I'll come back to you in just a second. So the first question, I have a similar follow-up question to using noisy data. How is the query generated? As an example of mad cow disease, wouldn't it affect the distance calculation given if you have a noisy query?
That is true. So that's a possibility, but let me. So for example, here I said I'm going to use this blue motif as the query, but I could actually do a few other things. So I would probably be better off not using the blue motif, but the average of the blue and the red, because the average is going to be smoother than either one of those two things. And I could do some other post-processing. I could actually say, you know what, this is 1 second, would probably be better if I cut off the head or the tail, make it a bit shorter, more specific. So I can do some manual data editing, manual cleaning. I can run a smoothing function or a spline function, and so forth. So I have, of course, kind of done this very high level and glossed over some things. You can put in domain knowledge, you can play around a little bit. But by definition, motifs are very highly conserved. So they tend not to find the noisy things. You tend to find the most typical things, almost by definition.
Great. Next question. One of the main component of distance or matrix profile is query. How do you identify the correct query? How much time is generally spent in identifying it if done in automated manner?
So you can find queries in multiple ways. Sometimes the query is given to you by a domain expert. You can simply ask the domain expert to sit down, look at video, or look at what it might be, and he will say, this is dust bathing. It starts here and it ends here. And you can take that dust bathing behavior, or what it might be, and use that as a query. But very often I find the queries automatically with the Metis profile. And simply the fact is, if a behavior is conserved, there must be a biological reason for that. In evolution, it might be. And so the most conserved behaviors tend to be the most important, the most typical behaviors. For a chicken, the most conserved behavior is pecking. They peck 40,000 times a day, right? And so that tends to be the best motif, and you can pull that motif out. So motif discovery suggests queries most of the time, but you can find queries any way you want with out-of-band information or hypotheses and so forth. You can do transfer learning. So I learned queries from chickens, and they almost all work for turkeys. Not unsurprisingly, turkeys and chickens are quite related. So whether your queries from a cow works between different cows, Daisy to Mildred, probably does. They look from different breeds of cows, it probably does. Does it go from cows to buffaloes? It might. Cows to pigs, maybe not at that point, right? So there is some domain knowledge you can push into this, basically.
Great. And I'll take one more before going to Mark. This is from Owen. Does the matrix profile replace shapelet mining, or do we still need a semantically meaningful shapelet or subsequence in the first place?
They're highly related. I guess the matrix profile can be used to find shapelets. It's one of the ways. But to be clear, a shapelet is simply a subsequence that's known to be overrepresented in one class and not another. My favorite example is if you want to classify Harry Potter American editions and Harry Potter UK editions, then the word trouser is a shapelet, because the word trouser appears in the UK edition, but not in the American edition where it's pants. Okay, so that would be a shapelet in that world. And there are shapelets that exist actually in our bovine or other animal world, which are patterns that exist more commonly in males and females, or in sheep than goats and so forth. So we can actually use the matrix profile to find shapelets, and we can find shapelets with other techniques too. Okay,
Mark, you're up.
Well, I thought that was really good. It's very cool talk. Enjoyed it, Eamonn. Mentioning trouser reminds me of a specific Irish thing that people, when they take bribes here, it's called trousering the money, right?
There you go.
So that will pop up in a different, but I guess I like a lot of the questions, people are sort of focusing a bit on the query, because I guess when it's obvious, it's sort of nice, and there may be good intuitive reasons for picking certain things to start from. But there's still, surely, is always the worry, about whether you have the optimal query to start with, because otherwise I can imagine this could get quite circular, because you'll find what you set out to see, as it were, if you start with a given query.
Surely, but look at this query here. All I did was look at the entire data set with no annotation. I said, show me things that are conserved. Yeah. Now, it might have been that the most conserved thing is kind of boring, like what we might call a stop word in text processing. And I might have to actually just delete that. But eventually I find this thing, and it's so nicely conserved. And I haven't shown this actually, but I looked to see actually if it occurs again to the left and to the right. And it really was highly conserved. So I know it's probably causally conserved. There's something in the world or in the cow's brain that's making that happen.
Okay.
And I kind of visually noticed that happens mostly in ruminating. So I had a pretty good intuition this is going to be a good query. And I continued on, and it worked well. But you are right. It might be actually that the behavior could be polymorphic. If I have a semantic region called, Eamonn exercising, there actually could be two parts of it. It could be walking and running. And one query will only do one of those, not both. I need to actually have a more general rule that says, if you see this, or you see this, it's Eamonn exercising. So there's many things you can do, more general than this. But
I guess I was thinking of stock markets and that, which are very noisy. And of course, the underlying model can be changing sort of too. And it's often like, there's these people that talk about visual investing. And my experience of that is that people say you look for a particular shape, but actually that shape can be interpreted in lots of different ways, sort of,
Yeah, I'm a big skeptic about that. That's called technical analysis. I'm a big skeptic about that. But in the physical world of physical devices or logical devices.
Yeah, there should be a structure, shouldn't there? Yeah.
Yeah.
Yeah. Okay, good. Thanks.
All right, Brian, you're up.
Thanks, Cheryl. And as Cheryl predicted, Eamon, I thought that talk was brilliant. So thanks very much for sharing it with us. I was going to ask a question just about you talked about normalizing and focusing in on shape. I know when we work in time series, you inevitably end up in a discussion about whether we should normalize or not normalize. And I wonder, do you have experience where the magnitude is an important part of the signal within the data? And are there modifications to things like the matrix profile in those instances that might be useful? Or do you think that's wrong and really we should always normalize and comes down to shape?
And that's a great question. Occasionally magnitude is useful, but when it is useful, the shape tends not to be. Let me start by normalize. For example, look at these cows here. When I looked at all this data, I think sometimes the color on some cow, it shifts to the left on one cow, it shifts to the right on the other cow. And now I cannot compare between those two cows unless I normalize, right? A trivial, maybe an inch to the left, inch to the right will completely dominate my analysis unless I remove it by normalization. And so the real world tends to be very brittle like that, and you have to normalize. So there are a handful of cases when magnitudes are important and I don't normalize. And actually, matrix profile and mass both support that if you want to. You can just triple those very easily. But again, my claim is 99% of the time, at least, you should be normalizing. Okay, brilliant. That's really interesting. And then I guess if there is time for another question, I'll ask the deep learning one. And as you were talking about the matrix profile and a grammar and a sequence, it struck me that a sort of an obvious downstream approach would be, well, throw that into a transformer model and maybe good things would come out of that. Do you remain sort of skeptical about deep learning for that, or do you think there might be mileage in it? People actually have done that, but in most cases, I think the result is no better to use matrix profile directly. I want to make it clear, I'm not a deep learning skeptic in general, right? It's an incredible tool for all kinds of things, images and things like that. But in the limited case of time series, I think it's special because it's so simple, actually, in some sense. There's no real evidence that has added much value. But I would say, actually, maybe at the higher level, once you have these motifs are basically now words, like words in English. And you can certainly use models on English words, obviously, right? So maybe possible to actually use matrix profile, find the vocabulary, give the vocabulary to these other deep learning models, and do something very, very cool. That I think could be explored and be very interesting. Okay, brilliant. Thanks for that.
I have another question from Owen. Has the matrix profile been extended beyond time series data to image data?
Not really per se. I mean, obviously the data can come from video. Actually, here's an example of a video of a young lady with a gun, and we played that time series extracted from it. But the matrix profile directly on the pixels, no. I'm not sure what that would mean or how it would work. I'd be interested to think about. Okay. And Bhaskar wants to know, do you have any intuition about rocket or mini rocket classifiers? What makes them work better than another? I have a limited intuition about those. They do work very, very well. So time classification is very interesting because a baseline is Euclidean distance nearest neighbor, which takes 5 lines of code and works quite well. And there are hundreds of papers that have more complicated algorithms that can do better. What's surprising though is you have to have an algorithm that's 1,000 times more complicated to get 1 or 2 more percent. And of course, 1 or 2 more percent can be very useful, but the trade-off is really quite surprising. Euclidean distance gets you almost all the way in almost every data set. And if that extra complexity is really warranted is not very clear. And one other comment I want to make is, this is kind of a bit skeptical here, is all these algorithms have been demonstrated on the UCR archive, which is an archive I've invented over the last 20 years. And there's a big problem that people actually overfit into that archive, which is a bit of a contrived archive. It's not a real proxy for the real world. And so when you take these rockets and so forth and you apply them to cows and pigs and chickens, they tend to actually do much worse than matrix profile and other techniques, right? It's because they're kind of optimized for these simple contrived data sets, and the real problems that we discussed earlier of noise and so forth really confound and confuse them. Okay, are there any more questions from our panelists? Mark?
Just a quickie. It occurred to me when you're talking about features, you seem to be using them fairly directly. Have people looked at all about at feature weighting within that context, the way they do with normal features, as it were?
Yeah, actually, I've been doing it myself as a student lately. Looking at catch 22, and for most problems, actually, a good feature subset is better than all the features. And there's this kind of paradox. If you have labels, you can kind of quickly learn the best feature subset. If you don't have the labels, it's hard to learn a feature subset. So yeah, there is some kind of black magic to these features. So one trick you can do is transfer learning. If you learn a good set of features for Bob and Joe has a similar domain, they probably will generalize, but you can never guarantee, because maybe Bob is much taller or shorter or whatever it might be. So there is a bit of black magic in this feature weighting, and all the tricks you already know, PCA and blah, 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 regression, probably apply to be used in that space.
Okay, thanks.
We have, Chang Hong has asked 2 questions. He's getting in right before the end. Is it necessary, and how do you consider overlap when extracting catch-22 features from subsequences in time series?
I typically simply just take every subsequence with overlap, just click by one, click by one, click by one, and get all of them. Because computationally, if you're efficient, it doesn't hurt. And there's always a danger if you skip, 1 hour, 1 hour, 1 hour, that the real patterns are in the overlap between. So I tend to extract all possible subsequences, which is computationally efficient and reasonable to do.
When using decision rules in matrix profile for determining the vocabulary, is it more significant to have the multiple occurrences of time series motifs or to have lower values of time series motifs?
So I should actually say, let me go back to that example briefly. The matrix profile's job is to find these motifs. This is kind of just an example. What I could do here, but you actually can do anything you want here. You can make the rules be complicated or simple. You can make them probabilistic rules or logical rules and so forth. That's kind of a downstream application, and that's your choice. In a sense, my job, I think, is to get you these beautiful motifs, and you can do what you want to do. So I'm not being evasive, but I'll make sure that you understand that this is a very flexible approach. The key part is to get these atomic units to be able to reason with in the first place. Okay.
And I think it's only fair to give Brian the final question of the session.
Thanks for that, Cheryl. And Eamonn, just if there was time, one sort of, I suppose, meta question about the matrix profile. And one thing we've discussed a little bit about in our reading groups, in our research group, is the sort of naming convention and a very specific approach to make those papers a set called Matrix Profile 1, Matrix Profile 2, and so on. Yeah. When you look back at now and kind of reflect on that, do you think that's do you have any reflections on sort of that process and that approach to doing that, and sort of positives and negatives that might have come from it?
Yeah, it is unique. I think actually no one else has ever done this, as far as I know. Yeah. There is this kind of meta question of Brandon, and wants to be visible and get promotions and tenure and grants and all other good stuff. And I think actually it was rare, but this was a logically coherent set of tools. So I think actually it made sense to do this. So partly it's kind of just a little bit fun to kind of do this, but I think it also really does actually make sense in this case. In this limited case, it makes sense. I'm going to give a tutorial, I'm hoping, later this year on how to brand as an academic. So if we actually have any young professors or young grad students who are kind of interested in building a career, I do have some hints on how to build your H-index, how to build a brand of identity that I think actually can be useful. I have a friend, Jeff Webb, who's an unbelievably smart guy in Australia. And I think he kind of recognizes that he didn't brand himself very well, and that had he done things differently, he would have been much more famous and much more, influential. So it's kind of a weird thing in academia, as you probably know, right? You can kind of be a bit aggressive and bold. Doesn't hurt in academia, right? It's actually probably helpful. And this is kind of an example of this. I kind of wanted the world to say, look at this. This is actually worth your time and attention. So hopefully that makes sense.
That's brilliant. Yeah, thanks very much for that.
Well, look, Eamonn, thank you very much. I know all of our computer scientists have really learned a lot in this session, and we really appreciate you taking the time to come and talk to us. And Yeah, thank you for answering all the questions, the many questions. And thanks everyone for coming to the AI and Ag Masterclass.
Oh, thank you all. Just quickly, any questions, follow up offline. And even in Southern California, I have a very nice lab in Riverside. Come visit me. I'll show you the chickens and we can chat.
Thank you so much.